Welcome back, students. Welcome to biology classes. Today we are going to see 11th science biology and we are going to resume the chapter 13, that is photosynthesis and higher plants, right? We have already completed many topics and yet today we are going to see the most important topic of this chapter, and that is Calvin cycle, right? This Calvin cycle is also known as C3 cycle, as well as also it is known as reductive pentose sugar phosphate pathway, right? So this can be asked in different ways. Now, as the Melvin, Kelvin, Benson and their colleagues, right? They were the ones to discover, as the main scientist was Melvin Kelvin, right? For this reason, after the name of the scientist, this cycle has been named, right? That is the Kelvin cycle. Now, Melvin Kelvin, Benson and their colleagues, what they discovered is that the first CO2, that is the first carbon fixation product, is a 3-carbon organic compound, right? And that is 3-phosphoglyceric acid, or we can simply say as PGA, right? So as the first carbon fixation product was a 3-carbon compound, for this reason, this cycle is also known as C3 cycle. But later on, what happened? A new compound was been discovered, which was containing 4-carbon, right? And for this reason, that 4-carbon compound was a oxaloacetic acid, which is simply said as AAO. Now, on the basis of number of carbon atoms in the first stable product, they are named as C3 and C4 pathway. So, if three carbon stable product is there, then it is known as C3 pathway. And if a 4-carbon stable compound is present, then it is known as C4 pathway, right? We are going to see the difference between both of them. But before going to see the difference, first we will see about the exact Calvin cycle. So the Calvin cycle is described under 3 stages, right? So there are 3 steps which are present in the Calvin cycle. The first is the carboxylation, second is the reduction, and the third is the regeneration, right? So we are going to see the 3 of the steps in detail. First is the carboxylation. So in the carboxylation, what happens is the fixation of CO2 takes place. That is, the fixation of carbon dioxide takes place into 3-phosphoglyceric acid, and that is 3-PGA. As 3 carbon is present, for this reason 3 PGA is present, right? So the first step of the Calvin cycle is fixation of CO2, right? Now, carboxylation of RUBP occurs in presence of enzymes. So carboxylation of RUBP, that means there is presence of RUBP, right? And this RUBP is what? Ribulose bisphosphate, right? So the carboxylation of RUBP takes place, that is the fixation of CO2 takes place, right? And what happens? In the presence of RUBP carboxylase, that is Rubisco enzyme, right? This leads to the formation of 2 molecules of 3-PGA. So you can see this is the figure of Calvin cycle. Here this is RUBP. In the presence of the RUBP carboxylase enzyme, the fixation of RUBP takes place, and here the carbon dioxide is been fixed, and for this reason, upon carboxylation which is taking place, this RUBP is fixed to 2 molecules of 3-PGA, right? So this is about the first step, and that is carboxylation. So in the second step, the reduction is taking place. So reduction is series of reaction that leads to formation of glucose, right? So when the reduction of this 3-phosphoglyceric acid takes place, at that time, what is formed? The glucose is formed, right? Now, 2 molecules of ATP and 2 molecules of NADPH are required for reduction of 1 molecule of CO2. So if we are talking about 1 molecule of CO2, then it needs 2 molecules of ATP and 2 molecules of NADPH, right? So for the reduction of 1 molecule of CO2, it needs 2 ATP and 2 NADPH, right? And 6 turns of these cycles are required for removal of 1 molecule of glucose. Now, as you know that glucose, the molecular formula of glucose is C6H12O6, right? So C6, that means you need 6 carbon compounds, right? That is 6 carbon atoms should be present, right? For this reason, there should be fixation of 6 CO2 molecules. So in turn, if for one CO2 molecule, if it needs 2 ATP and 2 NADPH, then for 6 turns of cycles, right? Once the sixth turn or sixth time, if the CO2 is being fixed at that time, one molecule of glucose is formed, right? So here you can see over here that from this 3-phosphoglycerate, the reduction is taking place, and for this reason the ATP is utilized, right? So the ATP gets converted to ADP and the inorganic phosphate. Also the NADPH is getting reduced to NADP. So the reduction over here is taking place, and this 3-phosphoglycerate, or we can see the 3-PGA, this gets converted to the sugar, or we can say the glucose, and that is sucrose or starch, right? So that will be a triose phosphate, right? So this is about the second step of the Calvin cycle, and that is reduction. As the reduction is taking place, for this reason, the name of the step is reduction. The third and the last step of Calvin cycle is regeneration, right? So regeneration is what? To generate something again, right? So here also the same thing takes place. Here also the generation of RUBP molecules is taking place. And for why it is again forming this RUBP? In order to continue this cycle for many times, right? So as the cycle does not stop, but it the continuation of cycle is carried out, for this reason, again from the triose phosphate, again the RUBP molecules are formed. So again this RUBP molecules are again going to fix with the help of the Rubisco enzyme, and they are going to convert to 3-PGA, and this way the cycle continues, right? But for this conversion or for this regeneration of RUBP molecules, this process requires one molecule of ATP. So one molecule of ATP is being utilized, and it is converted, it is reduced to ADP. So upon utilizing the energy, the RUBP is regenerated. So this is the third step of Calvin cycle, and that is regeneration, right? So you need to draw this figure, and you need to memorize this figure, because the whole cycle you can explain from this figure, right? So this is one of the most important topic which is regularly asked. Now for every molecules of CO2 entering into the Calvin cycle, 3 molecules of ATP and 2 molecules of NADPH is required. Now let's calculate this, right? So you can see over here is, for one carbon molecule, right, for every, like for one carbon molecule, it needs 3 ATP molecules. Why? Because 2 over here and 1 over here, right? So this way 3 molecules of ATP and 2 molecules of NADPH is required, right? So as you know that here 2 molecules of ATP were used and 2 molecules of NADPH were used. So 2 over here, and this is the third one. So this comes to 3 ATP and 2 NADPH, right? So this way for one CO2 molecule, it needs 3 ATP and 2 NADPH, right? Now to make one molecule of glucose, 6 turns of cycle is completed. So total energy molecule which is required is, you can see this table. This table says for C, in, that means how much enter and how much is entering, right? So 6 CO2 is entering. So now 6 CO2, that comes to one molecule of glucose, right? But over here, if 6 CO2 is entering, right, then one glucose molecule is formed, right? Same way, here you can see that 18 ATP, right? Why it is 18? Because 3 molecules. So 6 into 3, that comes to 18. So same way, the how many ATPs are going to enter? 18 ATP. And how much ADPs are going to release? 18 ADP. Sorry, this 18 is sent over to here, but here it will be 18 ADP, right? The third, 12 NAD, because 6 into 2, that comes to 12. So 12 NADPH is entering and 12 NADP is removed out, right? So this is the calculation for the 6 molecules of CO2, which is going to form 1 molecule of glucose. So this is all about the first topic and today's first topic, and that is the Calvin cycle. So the next topic is C4 pathway, which is also known as Hertzsprung pathway, right? And this pathway, as the first 4 carbon compound is found as the stable compound in this cycle. For this reason, this pathway is known as C4 pathway. Now, this pathway was worked out by Hatch and Slack in 1965 to 1967, right? So this pathway was being carried out by Hatch and Slack, right? So as Hatch and Slack, they were the ones to explain or derive this pathway. For this reason, the name of this pathway is also known as Hatch-Slack pathway, and mainly operational in plants which are growing in dry tropical regions like maize, sugarcane, sorghum, etc. So normally this type of pathway is seen in the dry tropical regions, and the crops like maize, sugarcane, or sorghum, etc. Right? Now, in this pathway, the first stable product is a 4-carbon compound, that is oxaloacetic acid. So, as the first stable compound of this pathway is oxaloacetic acid, for this reason, this pathway is known as C4 pathway. And the C4 plants, they are showing Krebs anatomy, right? So for that, you need to understand that what exactly Krebs anatomy is. So the Krebs anatomy is exactly what? That the vascular bundles are surrounded by bundle sheath cells in wreath-like manner. So when the vascular bundles are surrounded by a specialized cells which are known as bundle sheath cells, then this type of anatomy is known as Krebs anatomy, right? Vascular bundles is going to contain 2 types of tissues. The first is the xylem and the second one is the phloem. So when the vascular bundles, which is made up of the xylem and phloem, when there is presence of bundle sheath cells around the vascular bundle, at that time, that type of anatomy is known as Krebs anatomy. Now, this Krebs anatomy, it is been characterized by the large number of chloroplasts, thick wall impervious to gases, and absence of intercellular spaces. Now, this Krebs anatomy, they show larger number of chloroplasts which are present. Also, their thick wall is impervious to gases. That is, they are not permeable, or they are not allowing all the types of gases and everything to pass, right? They allow only few selected things only to pass. And also there is the absence of the intercellular spaces between them. Now, the primary carbon dioxide acceptor, that is CO2 acceptor, is a 3-carbon molecule, that is phosphoenol pyruvate. So the first one who is going to accept the CO2, that is phosphoenol pyruvate. And this phosphoenol pyruvate, it is present in the mesophyll cell. So in C4 pathway, you are going to see 2 types of cell. The first is the bundle sheath cell and the second is the mesophyll cell, right? Now, the first carbon molecule, it is been accepted by phosphoenol pyruvate, and this phosphoenol pyruvate, it is present in the mesophyll cells, right? And the enzyme which is involved, right, that is PEP carboxylase, that is phosphoenol pyruvate carboxylase, right? So as it is going to act on the phosphoenol pyruvate, right? For this reason, the enzyme which is going to fix it, that is the PEP carboxylase, right? And upon the fixation, the oxaloacetic acid which is formed, right? So that is formed in the mesophyll cells, and this is going to be a 4-carbon compound, unlike malic acid or aspartic acid. And this is transported to bundle sheath cells. So none another, when the phosphoenol pyruvate accept the carbon dioxide and fix it with the help of the PEP carboxylase enzyme, it is going to convert it into a 4-carbon compound which is known as oxaloacetic acid, right? And this oxaloacetic acid, is transported from the mesophyll cell to the bundle sheath cells. Now, what happens in the bundle sheath cells? So, in bundle sheath cells, this oxaloacetic acid, it is broken down into carbon dioxide and a 3-carbon molecule, right? So in the bundle sheath cells, what happens? It is broken. So 4 carbon molecules are there. So from that 4 carbon molecules, 1 carbon dioxide is released and a 3 carbon molecule is formed. This 3 carbon molecule is written back to the mesophyll cells, and again it forms the phosphoenol pyruvate. And this way the whole cycle or the whole pathway continues, right? Now the CO2 molecules which are released, right? So over here from the 4 molecules, 3 carbon compound which was formed, that is phosphoenol pyruvate. But also there was a removal of 1 carbon dioxide molecule. So this carbon dioxide molecule which is released in the bundle sheath cells, this carbon dioxide, it enters inside the Calvin cycle, where with the presence of the Rubisco enzyme, it forms the sugar. So now we will see the figure of this pathway, right? So as you can see over here, that this is the phosphoenol pyruvate. This is first we will see that the upper cell, this is the mesophyll cell, and the lower cell, this is the bundle sheath cell, right? So starting with the first, that is the phosphoenol pyruvate, right? So this phosphoenol pyruvate, this PEP, in the presence of PEP carboxylase enzyme, right? It fixes the carbon dioxide, right? And it gets converted. This phosphoenol pyruvate is already a 3-carbon compound. But when another 1 carbon is being added, at that time it is getting converted to a 4-carbon compound, and that is oxaloacetic acid, right? So that is C4 acid. Now this C4 acid, that is oxaloacetic acid, it is entering inside the bundle sheath cell from the mesophyll cell. So once the oxaloacetic acid is formed, it transports itself from the mesophyll cell to the bundle sheath cells. Now when in the bundle sheath cells, right, the decarboxylation takes place, right? And here there will be splitting of the carbon molecules, and 1 carbon dioxide is released out, and the 3 carbon compound which is going to form C3 acid, and that is going to regenerate the phosphoenol pyruvate. So as you can see, this is releasing out CO1 molecule of carbon, that is in the form of carbon dioxide, and the left 3 carbon molecule, this is going to form a molecule which is going to pass from the bundle sheath cell to the mesophyll cell. So here you can see the C3 acid is passing or transporting itself from the bundle sheath cell to the mesophyll cell. So now this C3 acid has the ability to regenerate the phosphoenol pyruvate. So again, when the phosphoenol pyruvate is formed, again the carboxylation takes place, that is the fixation, and once CO2 molecule is been added into this, and it is going to form C4. Again it is going to enter bundle sheath cell, again decarboxylation takes place, again C3, again, and the cycle continues. And what happens with the one released carbon dioxide? This enters inside the Calvin cycle. So this way, as you can see, the Calvin cycle is a dark reaction, right? So that is occurring in dark. So here, as this cells, right, as the Calvin cycle, this carbon dioxide cannot be directly uptaken by the like the vascular bundles. For this reason, what happens is this cycle is not directly getting from the atmosphere, but the atmospheric CO2 is this way passed to the Calvin cycle, and this way the whole cycle runs, right? So this is all about the C4 pathway, or which is also known as H/slack pathway. So this is the difference between the C3 pathway and the C4 pathway, right? So in C3 pathway, as it is the first carbon compound is a 3-carbon compound which is stable in nature. For this reason, it was said to be C3 pathway, as here in the Hertzsprung pathway, as the first carbon compound is a 4-carbon compound which is stable in nature. For this reason, this pathway is known as C4 pathway, right? And the major difference between both of them is that C3 plants does not show Krebs anatomy, while the C4 pathway shows the Krebs anatomy, right? And what exactly Krebs anatomy is? So Krebs anatomy is the vascular bundles. They are being surrounded by a specialized cells which are known as bundle sheath cells. So I hope you are very clear with today's topic, and I hope to see you again. Thank you.